Hello everyone, this is Peter Cooper from the NCBI. Welcome to today's webinar on Standalone BLAST 2.9.0 and information relating to the database 5 format. So this has various titles. A shorter one is using organism. We're going to talk mainly about using the organism information with Standalone BLAST 2.9.0 and the BLAST database 5 format. This is really a webinar that I've already given almost a year ago, but we wanted to update you on some things about BLAST DB5 and just to remind everybody and to talk to those who didn't hear us the first time about what you can do with BLAST DB5. So what we're going to do today is give you a little bit of the motivation behind why we made these changes, talk about some of the features, show you how to limit your search by taxonomy, show you that you can search sequences by accession faster, and we'll show you how to retrieve sequences by taxonomy from a BLAST database. I think it's useful to understand why we made these changes. And the main reason that we did it is to prepare for changes that are coming to sequence identifiers. We're moving to systems that use accession-only sequence identifiers rather than the integer GIs or the integer IDs, which are called GIs. I'll come back to that in a few minutes. There's also changes in the protein chain identifiers coming from PDB. Those are the proteins that are coming from the structure database. That mean we need to make some changes in the database. So that's all very well and good, and it sounds a lot like we're doing this because we have to do it for our own purposes. But you get things out of this. One of the things that you get is something that I think people have wanted for a really long time, and that's to have the taxonomic information baked into the BLAST databases. These make your organism searches much easier. The database system that we're using, LMDB, enables faster lookup of accessions and other string identifiers. You will also get access to proteins from GI-less projects. We'll talk more about that in a couple of minutes. We're now producing proteins that do not have GIs, and you can't see those in the old BLAST databases. And also, once we get the new PDB records with the long chain IDs, you'll be able to access those in the BLAST databases. So just let me point out something to you. All of our software relies on these integer identifiers. There are various ones for various databases. The ones for sequence databases are called GI numbers, and they're these integers. And all of our systems have traditionally depended on these integer identifiers. These are basically ints in terms of C++ programming language. So there's a limit, depending upon whether they're signed or unsigned, to 2 to 4 billion. The thing that I want to point out to you is that we're already at about 1 billion GIs. So that means that we're going to run out. And so this will be a problem, meaning we will have to rewrite a lot of our software. And so we're going to move away from using GIs to using accession.versions instead. And because the number of GIs we have left is somewhat limited, we're kind of rationing those. So for some projects, in particular, a lot of the whole genome shotgun projects that are coming in now, that we do make proteins for them, we've stopped assigning GI numbers to those. And so there's a whole bunch of these that you can see here in the sequence set browser. They're nucleotide and protein sequences that do not have GI numbers. What this means is that you can't find these things by searching in nucleotide and protein web resources by using ordinary queries. You can find them by their identifiers and pull them out. And these things, the proteins in particular, are not going to be present in the V4 BLAST database, the old version of the BLAST database. Just to point out, many of these proteins are coming from the Pathogen Detection Project, which is this collaborative project between a number of government agencies that's involved in identifying and analyzing foodborne illness outbreaks. So this is one of the major sources of GI-less proteins. And these are important proteins, of course, because these organisms are important and their proteins are important. So you need to be able to have access to those, and that's why you need to move to the new BLAST version and the new BLAST database version. What's been up for a while at NCBI is sort of a not quite fully DB version 5 database, which means that it does have GI numbers in it. In order to work with the fully GI-less database, you're going to need to upgrade both your BLAST binary to 2.9.0 and to get the new DB version 5 database. The executables for 2.9.0 are available in the usual place on the BLAST FTP directory, in the latest directory, in the latest link in the FTP directory there for executables. 2.9 will still work with version 4 databases. I just want to point out that we will stop updating the version 4 databases eventually, and I think we're thinking about stopping after September of 2019. In order to work with the newest v5 databases, which are not yet on the FTP site, you will need to upgrade to 2.9.0. The updated GI list databases for v5 should be on the FTP site. We're thinking May 20th. And just, I have a little footnote here, although it's not something that I want to advertise as a great thing. The side effect of that we're using these on the web right now is that you'll notice that you can't use entree limits anymore for protein images. The organism limits are still there, and that's the thing that most people need. And speaking of that, the thing that I always tell people when they do a blast search on the web is the most important thing you can do is to limit by organism. And you can do that by typing an organism name in the bottom here of the blast form where it says organism to either include or exclude various taxa. On standalone blasts, the way you would manage this in the past has been traditionally by GI lists. And that's not going to work anymore with the version 5 databases. So in order to do this, you can do some other things that don't require using GIs. One is to use a seek ID list. Seek ID lists can be accessions, so you can use that accession.version, a list of those to limit to a particular set of sequences in the database. You can also exclude things using a negative seek ID list. Again, this can be a list of accessions that you don't want to see. And then there's a whole set of things that have to do with the baked-in taxonomy that's in the database. So you can search for a particular tax ID or a small list of them that you can pass as an argument. Or you can pass them as a negative argument, so you want to exclude certain groups of organisms, or you can upload a file of tax IDs to either include or exclude from your search. These things work on the search programs for BLAST DB-CMD, which is the program that you use to extract things from a BLAST database. You can retrieve tax IDs, sequences for particular tax IDs, or a list of tax IDs directly using BLAST DB-CMD. You can also, looking at any of the databases that are in v5 format, you can get tax info. It tells you what taxa are in there and how many sequences each taxa has. So how do you get a list of tax IDs? You can do that on the web fairly easily. So you can just go to the taxonomy database here at NCBI, and you can search, for example, a set of organisms. So I'm getting all the tax IDs for green plants in this particular case, and download that. And I could even do an exclusion if I wanted to get a particular set of tax IDs, in this case for green plants without the monocots in them. So you can create a file and download that and use that to limit your search. In this case, this is about 209,000 tax IDs, which really, it's not too bad. You can download that in a few seconds. You can also do it from the convenience of your command line with a shell script that comes with the BLAST release now, and it's called get species tax ID, and it's a shell program. You can either input a name to get back a tax ID, or you can input a tax ID to get all the tax IDs concluded in the taxon. This uses our eDirect package, so you have to have that installed, and for that reason it only works for Unix, Linux, and SIGWin only at this point. So you don't have to use this to get those tax IDs. As I showed you, you can do them from the Entree system, but it's pretty convenient to do it. And so here are some sample command lines that that use these various new options that are available now. And we'll go over some of these on the command line here and directly. So I won't go over these by reading them aloud to you. I don't think that's very helpful, but the slides are there. And you'll find that if you look at the slides that I gave you, there's a couple of extra slides in there that I didn't use when I gave my talk today. Okay, so let's go back and we'll do some live demos, which is always exciting because they don't always work. Something may go wrong, but that's fine. We're going to look at a GI list protein just so you can see that these things really do exist. We'll get some tax IDs for soybean flowering plants. And actually, I think I changed that up. I think I used the P family instead of flowering plants because that was such a large set. And we'll show how to limit a BLAST-P search against NR to those particular taxa. We'll show you how to extract certain sequences from NR using the tax ID list. And we'll show you how to use a seek ID list that's been converted to a binary form so that it works much faster. So I'm going to escape out of my slides here. and I'm going to move over to a command line window. And what I'm going to do is switch back and forth because I can't type very well. So I'm just going to copy. I have the text file up on the FTP side. You could certainly look at it, and it's in your handouts there as a Word document. So you can see the command lines there if you want to. I'm just going to copy paste those when I need to so we can see them in action. Okay, so let me get a terminal window up here. So what I'm going to do is switch back and forth between this and my web browser. So the first thing I'm going to do is just to show you that there are GI list proteins in the database. And this is and what I'm doing is pointing at a copy of the NRB5 that's on our system here. And so this is just a typical use of BLAST dbcmd to get out the FASTA format for this particular protein. And this is one of these proteins that came from our pathogen detection project. And this is one of these drug resistance proteins from Salmonella enterica. Now, how do I know that that's a GI list protein? Well, one of the things I can try to do is just let's see if we can pull it out of the version 4 database. And I just pointed at the V4 here, and you can see that that doesn't work. That will prove it. And there's a couple other command lines that I won't waste your time with, but you can show that there really aren't any GIs in this database, in the real DB5 database, which DB version 5 database, which remember is not yet available on our FTP site. So let's go ahead and move on, and we'll do some of the basic things we talked about a minute ago. So we'll switch back and forth here a little bit. I'll go ahead and open a new tab over here. And what I'm going to do is do a search in, I'll go ahead and just load this directly, but this is what I could do in taxonomy to get tax IDs. So you can take any organism name, go to our taxonomy database. This is a species level tax ID, which is what you're going to need to work with the BLAST databases. And really, it's the leaf node tax ID. So I could have the tax ID is 3847. So I can easily find that on the web if I want to. I could also do it command line style if I want to. And I could also use the script that's provided with the BLAST database, the BLAST distribution. And we'll see that in just a minute. Notice that I can also get all the tax IDs for a particular taxon by going and searching for the P family, for example. And so that's the entree query that I need, P family and that subtree in square brackets. And it gives me 13,000 tax IDs. I could then save these to a file. I want to change this to tax ID list. And then I can create the file. And I've already done that, so that's on my little directory that I'm using for the webinar. Just to save time, I won't go ahead and download that. We can also do the same thing using the script. Let me go back over here. So here's our get species tax ID shell script. And just like many of our BLAST programs, you can just type help at the end of it to get options. This is a pretty simple script. The taxonomy ID is one of the arguments I can give it, or I can give it the name of the organism that I'm interested in. So let's say I want to do the P family, which is a fairly large group of plants. So that's the tax ID, and now I can use it to get all the tax IDs that I would need to make my list. The same thing that I did on the web a second ago. And what I'll do is I won't bother to redirect that to a file right now. I'll go ahead and just show you part of the output. And so you just get that long list of tax IDs In fact, that file is here, so I can just count how many are in there. And that's the same number that we had on the web, 13,176. Okay, let's do some other more interesting things now. Let's go ahead and limit our blast searches to a particular tax ID. And so let's search the NR database, and I've got a bunch of options here on the command line that are useful ones. We've got a particular kind of tabular output, and this will be searching only those tax IDs. This is the soybean tax ID, and I've got this Schizosaccharomyces protein Argo that I'm going to search against that with, and I'll go ahead and just put the output to the terminal, just because it'll be a little faster. Now this search depends on how many people are using my blast machine here as to how fast this is going to be. So let's give it a shot and see if it's fast. It was fast a few minutes ago. And if I get tired of waiting for it, I have the results available so we can look at them in a minute. I'll give that about 15 or 20 seconds, and then I'll go ahead and bail on it and show it to you. There it is. So that's the head, and the total output is a similar kind of thing, and it just goes on and on. But we have focused our search against NR only against proteins from glycine max, which is the soybean. Likewise, I can do the same thing for peas. In this case, I'm using the tax ID list. Let's just redirect the first 10 lines of that to the output. Let's see how fast this one is. This is a little bit more data. I'll give that about 20 seconds too, and if it doesn't get done, I'll just cancel it and show you the output that I've already saved. So that was pretty fast. Now, admittedly, I have a pretty machine with a lot of memory that I'm using. But again, we see other members of the pea family. In this case, the first few here are from the genus glycine, same as soybean, but it's a different species. So suppose we wanted to get out all the soybean proteins that are in our 5 database. We can easily do that here. So I'm using blast dbcmd, and this is one of the command lines that was in my slide. So I'm just going to tell it to give me all of the FASTA formatted sequences, and I'm not going to show you that whole output, but I have the entire file already done just to show you that this does work. There's the first couple of them right there. I can do exactly the same thing with the pea family. It doesn't really add any additional information, so I won't demo that one for you, but it's in the handout if you want to try it yourself. So the last thing that I want to show you is making an accession list and then using that to focus your search on a particular set of sequences. And what I'm going to get is the list for the human non-model ref-seqs. And I've got the entree query right there that you would use to get these. And this is actually a pretty fast way to get them. I'll just show you that on the web. So there's 53,000 of these. You can easily download these to a file. And what you want to do is to get your accession list. And I've already done that, so that list is on my little directory there. There's another way you can do it. You could also use eDirect to do this, and the eDirect command line is also in that handout. I'm not going to bother to run that. And so I could do eDirect in exactly the same way. So I have this file on here. This is human non-model accessions, and these are going to be the NP style reference sequences. So there's the first 10 of them. There's 50-some thousand in there. And then what I want to do is create a version 5 seq ID list. And this uses the BLAST utility that comes with BLAST. It's the BLASTDB alias tool. And it's going to make this binary seek ID list file that we're going to use to limit our search. Okay, and that file is there. There's my non-model seek ID list right there. And the last thing I want to do is just run that search with the same kind of options that we use for all the other ones. And again, I'll go ahead and put this to standard output instead of putting it in a file. Okay, so that worked just fine. So now let me just go ahead and go back to my slide set here. So we showed you that the improvements that we've done to last 2.9.0 and DB5 works faster with string lookups. It's much easier options for taxonomic filters. Works faster with accession lists. And remember that this is required for the true GIS databases that are coming soon. And I'll leave you with a few URLs here for things that we've talked about in the webinar and places to go for additional help. For example, our YouTube channel is up here, so that's where this recording is going to be. And I'm going to leave it open for a few minutes for questions. I realize that we're pretty much at time, but I can continue to answer questions if there's anything that. So Dave Arndt is here, and he's going to read me the questions.
Someone asked about RevSeq genome databases. When are they going to be in BLAST-DB version 5?
That's a good question for Christian to answer. So that means that the genome databases are not going to be available in version 5 for a while. Now the proteins associated with those are in the regular protein database, so those are going to be in there now.
Okay, another question is, what would you recommend for finding or narrowing down tax IDs when you are looking for an order of like animals, when the taxonomy listed in the database is not correct or doesn't seem to be correct for some of the entities when you do an alignment?
Yeah, I'm not sure I understand the question completely. I mean, one thing to say is whether or not our taxonomy is correct or not. What people think about the classification of organisms doesn't really matter so much because it's just what we use. So you would have to use the tax IDs that we use for that particular group of organisms. So that's all I can recommend, is that you're going to have to use our tax IDs because those are the ones that we've assigned to the sequences. Whether or not those are the true classification system is open for debate, but I'm not sure if that answered the question or not.
The next one is it possible to filter by type species or type strains?
Yes, that's possible to do by making an accession list of those things. I don't know that that's an option to search for the tax IDs in taxonomy, but it may well be. Yeah, I don't. That's a good question, and I'll tell you what. In the document, I'll look into that and answer that in the question and answer document. I know you can do the kind of search in protein, for example, to get sequences from type. And that's really just for efficiency, right, Christian? If we stored all the lineages, it would just be too much. It's just too many nodes in there. When we did this in the demo, we limited to the P family, for example, and you can do that but you do have to get all the leaf node tax IDs. And so anything that's in the questions pod, we will address in the document that's going to be provided in a few days. Okay, so thanks everybody for coming, and that's going to conclude the webinar for today. Nice talking with you, and we'll talk to you next time. Thank you.